Okay, so it's 10:01. Let's get started. Good morning, everyone. Welcome to this month membrane trafficking webinar sponsored by the Society of Chinese Bioscientists in America. And so I'm Yi Hongye from National Institutes of Health. Before I start introducing today's seminar speaker, I want to take just 1 minute to advertise a new conference sponsored by Cold Spring Harbor Asian Conferences. This will be held in Suzhou in March 10 to 14, China. And the topic is about molecular mechanisms and physiology of unconventional secretion, which is quite relevant to today's seminar. So this is a brand new meeting on this relatively underexplored field of membrane trafficking. And meeting is really designed to facilitate the science of young scientists. We have more than 20 short talk slots, which will be selected from submitted abstracts. And we also have fellowships that sponsor travel of promising young scientists to these conferences. And if you wish to get the fellowships, please apply early. And also we have pretty much awards for every poster presentations at these conferences. So really, this is going to be a very exciting meeting on this emerging new field of membrane trafficking. And I hope to see many of you at this in this beautiful city of Suzhou in China in 2025. So, and then with that, I'm gonna start by introducing today's seminar speaker, which is a young star, Ming Zhang, Associate Professor of Tsinghua University. Min received her PhD from the Chinese Institute of Biological Science in Chinese Academy of Science in 2011. And then she joined Dr. Randy Shackman's lab at UC Berkeley there to become a postdoc fellow in Randy's lab. And she started working on this topic of unconventional protein secretion and made some very important discoveries that allow her to establish her own lab in 2021 at Tsinghua University. And since then, she has made some similar discoveries, particularly reporting the involvement of this family of protein called TMED10 in unconventional protein secretion. And I'm very glad to have her here today to share with us her newest finding, focusing on how this TMED10 protein is regulated and this unconventional protein secretion mechanism, and how would that be relevant to human diseases. So, Min, the stage is yours. Thank you.
Thank you, Hong, for the introduction and giving me the opportunity to present my work. I'll share my screen. Okay. Okay. You see it? Okay. So, my lab.
Can you turn it to the presentation mode? Oh, yeah. Okay. Now it's it. Okay.
Yeah. So my lab mainly focuses on the molecular mechanism and the function of unconventional protein secretion. And today I'm going to share with you our recent progress about the protein translocation and the physiological function of unconventional protein secretion. So talking about protein secretion, I guess everyone is familiar with this conventional secretion pathway. In this pathway, most of these proteins, I think most of the proteins are secreted through this conventional secretion pathway. And these proteins, for example, insulin and IL-6, contain N-terminal signal sequence that could direct the protein to the ER, and then these proteins could be transported through the ER-Golgi membrane trafficking pathway for secretion. In contrast to conventional secretion, unconventional protein secretion refer to the secretion of proteins without the signal peptide and independent of ER and Golgi. So this unconventional secretion protein includes cytokine family protein, IL-1 family proteins, alarmin, S100, A8, A9, neurotoxic proteins, including tau and alpha-synuclein, galactins, and growth factors, including FGF1 and FGF2. Due to lack of the signal peptide, the secretion pathways for unconventional secretion are diversified. So far, there are two major secretion pathways. One pathway do not depend on the vesicle intermediates. In this pathway, proteins directly translocate across the plasma membrane. The other pathway depends on the vesicle intermediates, and this pathway is very similar to the conventional secretion pathway. However, since these proteins do not have the signal peptide, a key question in this pathway is that how can these proteins enter the membrane trafficking pathway? So in the past years, our lab tried to answer this question. And so far, we have identified a protein translocation pathway for vesicle-mediated unconventional protein secretion named TMAT10 channeled unconventional secretion, THU. The key of this pathway is the single transmembrane protein TMAT10, which localizes on the very important membrane compartment called the ERGIC. It is the intermediate compartment between ER and Golgi. And the working model for this pathway is that during secretion, the cytosolic cargo, for example, IL-1 beta, could get unfolded and then translocate to ERGIC. And at ERGIC, the cargo could associate with TMAT10 to stimulate the oligomerization of TMAT10 to form a protein channel. With the help of another chaperone protein, the cargo could translocate through the protein channel into the lumen of the ERGIC, and then the cargo could later be refolded and secreted by downstream trafficking pathways. So for THU, two questions are still not clear. One is what is the detailed mechanism of THU at the ERGIC? And the other is what is the physiological role of THU? So in the following slide, I'm going to show you two stories. The first is about the regulation of THU by ERGIC localized rep proteins. The second story will show you the physiological function of THU in host inflammation upon coronavirus infection. So for the first story, as reported, rep proteins play important roles in membrane trafficking, as shown in this picture. And regarding protein secretion, as reported by recent studies, Rep37 could regulate the conventional secretion of IL-6. Meanwhile, Rep8 and Rep5 have been reported to regulate unconventional secretion. So our question is whether rep proteins could also regulate THU. To determine this, we did a screening on the rep proteins by overexpressing them individually in our cell-based unconventional secretion analysis system. And we found that the presence of Rep5 and Rep8 could slightly enhance unconventional secretion, which is consistent with previous reports. And importantly, we found three reps, Rep1A, Rep1B, and 2A, could greatly promote unconventional secretion. And knocking down any of the rep proteins could greatly block the unconventional secretion of IL-1 beta in macrophage. However, knocking down any of the three reps had no effect on the conventional secretion of signal peptide containing GFP. And moreover, the presence of Golgi trafficking inhibitor BFA could not affect rep-regulated unconventional secretion, indicating that these three reps specifically regulate unconventional protein secretion. And from the imaging results, we found that these three rep proteins localizes on ERGIC, indicating that these three reps could regulate THU. So here I would like to also mention that the GTPase activity of rep proteins is required for its function in unconventional protein secretion. Regarding the rep proteins are small GTPases that could switch between the GDP inactive form and the GTP bound active form. We found that the presence of QL constitutively active mutant promotes secretion, whereas the SN dominant negative form of rep proteins could block unconventional secretion, and moreover, overexpression of a gap or knocking down the gap could block unconventional secretion. So indicating that the function of rep proteins in unconventional secretion requires GTP binding. So then we determined if rep protein regulated unconventional secretion depends on TMAT10. We found that in TMAT10 kill cells, rep regulated unconventional secretion was greatly blocked, as shown here. And in the contrary, knocking down any of the three reps could also inhibit TMAT10 mediated unconventional secretion, indicating that these three reps act interdependently with TMAT10 in unconventional protein secretion. And here we were also able to show that these three rep proteins could directly bind with TMAT10. So then we determined if rep proteins could promote the entry of a cargo into the vesicles. We first use a membrane fractionation experiment to separate to collect the membrane fraction. We found that in the presence of Rep1A or Rep1B, the amount of IL-1 beta in the membrane fraction was greatly enhanced. To our surprise, the presence of Rep2A had no effect on the level of IL-1 beta in the membrane fraction. And we further used the split GFP system to determine if rep proteins could facilitate cargo entry into the TMAT10 vesicle. The idea here was that the entry of cargo into the TMAT10 vesicle could cause GFP complementation, and this GFP signal could be detected by flow cytometry. Consistently, we found that the presence of Rep1A or Rep1B could greatly promote the GFP signal, whereas the presence of Rep2A has no effect on the GFP signal, indicating that Rep1 could facilitate cargo entry into the vesicle, whereas Rep2 could not. So indicating that Rep1 and Rep2 regulate unconventional protein secretion through different mechanisms. So in the following slides, I'll talk about Rep1 first and then talk about Rep2 in the later part. So regarding that, Rep1 could promote cargo entry into the vesicle. So two key steps for protein translocation in THU are TMAT10 oligomerization and protein translocation through the protein TMAT10 channel. So then we determined if Rep1 could regulate these two processes. By using cross-linking, we found that the presence of Rep1A or Rep1B could greatly promote the oligomerization of TMAT10, and knocking down Rep1A or Rep1B could block the formation of TMAT10 oligomers, so indicating that Rep1A and Rep1B could enhance TMAT10 oligomerization. And then we further used previously established in vitro TMAT10 protoliposome system to determine cargo translocation. The idea here was that the TMAT10 could mediate cargo translocation into this protoliposome. So if the cargo enters into the protoliposome, it could be detected by Western blot after proteinase K digestion. So here we saw that the presence of RAB1A or RAB1B could greatly promote TMA10-mediated unconventional protein, the cargo translocation in a GTP-dependent manner, indicated that these two RABs could enhance cargo translocation. So then we also, by using this the imaging, we found that in the presence of RAB1A or RAB1B, more cargo could co-localize with TMA10, indicating that RAB1A and RAB1B promotes unconventional protein secretion by enhancing cargo association with TMA10 and therefore could enhance TMA10 oligomerization and protein translocation. So here I would like to summarize the role of RAB1 protein in unconventional protein secretion. We found that RAB1A and RAB1B could promote protein translocation activity of TMA10. So how about RAB2? How does RAB2 promote unconventional protein secretion? Before talking about the role of RAB2, I would like to talk about the very important membrane compartment, ERGIC. ERGIC typically acts as the sorting stage between ER and Golgi, and it plays important roles in unconventional protein secretion. But according to our previous study, ERGIC is also required for unconventional protein secretion. So when question come out, how does ERGIC coordinate conventional and unconventional secretion? So interestingly, when we looked into the detailed subcellular localization of TMA10 at ERGIC by using SIM imaging, we found that TMA10 and the ERGIC marker, ERGIC53, are not completely overlapped, but even with more than 50% separation, you can see this 3D image. So this suggested that ERGIC is compartmentalized for conventional and unconventional secretion. And then we asked if this TMA10 positive region is positive for unconventional protein secretion. To determine this, we first determined the region of ERGIC where conventional and unconventional secretion cargoes entry using split GFP. Here we use ERGIC53 as a marker for conventional secretion and TMA10 delta gold, which do not have the translocation activity, as a standby for unconventional protein secretion. And we saw that the conventional secretion cargo L1 beta mainly complemented with TMA10 delta gold, whereas the conventional cargo L6 mainly complemented with ERGIC53. And moreover, by using the imaging, we saw that more L1 beta could co-localize with TMA10, and more L6 co-localizes with ERGIC53, indicating that conventional and unconventional secretion cargoes enters into different regions of ERGIC, and ERGIC is compartmentalized for conventional and unconventional secretion. And we further use the rush system to artificially mix these two regions together by using the strip avidin and the SBP. We found that when we mix them together, we saw an increase of the co-localization between TMA10 and ERGIC53, and we saw a greater inhibition of protein secretion. But the addition of biotin, which competes with strip avidin, the binding of strip avidin and TMA10 and SBP, this could restore the separation of TMA10 with ERGIC53 and also rescues the secretion. So this result indicating that regional separation of the ERGIC is required for unconventional protein secretion, and suggesting that TMAT10 positive region is positive for unconventional protein secretion. And we further used the claim to visualize the morphology of the objects in conventional and unconventional secretion. Here, in yellow regions, with both ergic and TMAT10 localization, where conventional secretion occurs, we saw vesicular compartments adjacent to stacked Cargy-like structures. In contrast, where within the TMAT10 single positive regions, which are likely associated with unconventional secretion, we saw tubular vesicular structures more distant from the Golgi stack. So this result further confirmed that ergic is compartmentalized for unconventional and conventional secretion. And then we also used the imaging to determine whether separated TMAT10 co-localizes with the markers from other organelles. Surprisingly, we found that the separated TMAT10 do not co-localize with the markers of Golgi, ER-axis site, COP1, COP2, or endosomes. And interestingly, the separated TMAT10 partially co-localizes with RAP2A, indicating that RAP2A maybe regulates ergic compartmentalization. So here we saw that in the presence of RAP2A, more TMAT10 separated from ergic 53, indicating that RAP2A facilitates ergic compartmentalization. And we also used stone imaging to quantify the separation of TMAT10 from ergic 53. We saw in another cell line, called 7 cell line, we saw that in the presence of, when we knocked down RAP2A, could block TMAT10 and separation from ergic 53, and the re-expression of RAP2A could restore the separation. So, and we further also identified microtubule associated motor protein, kinesin 5B, that is effector protein of RAP2A. Knocking down kinesin 5B could block unconventional secretion and also block the TMAT10 separation from ergic 53. And we also, by using the co-IP and the imaging, we found that RAP2A is required for the association of kinesin 5B with TMAT10. So indicating that kinesin 5B is required for the RAP2A regulated ergic compartmentalization. So here I would like to summarize the role of these 3 RAPs in unconventional protein secretion. In this part of work, we show how cargo translocation is activated and how conventional and unconventional secretion pathways are coordinated within the ergic. We found these 3 RAPs regulate unconventional protein secretion through different mechanisms. RAP1 could promote the protein translocation activity of TMAT10. In contrast, RAP2A could collaborate with kinesin 5B to facilitate the formation of specific ergic region for unconventional protein secretion. Okay, so next I will talk about briefly introduce the second story about we have identified coronavirus envelope protein as a regulator for THU, and it affects host inflammation during coronavirus infection. So according to our previous study, we found that THU could regulate the secretion of a couple of proteins includes the very important cytokine family proteins, the IL-1 family proteins, therefore indicating that THU may play important roles in immune responses. So, and the outbreak of COVID-19 had caught our attention. As reported, the cytokine storm is the major mechanism accounting for deaths by COVID-19 infection. And from the literature, we found that, interestingly, multiple inflammatory factors, including the IL-1 family proteins, S100A8, A9, alarmins, annexin A1 are released through unconventional secretion. And coincidentally, as reported, the example of coronavirus occurs on ERGIC. So this indicated that there may be a relationship between coronavirus infection and unconventional protein secretion. And moreover, even after the release of the virus, the viral proteins remain in the cell, indicating that these proteins may have other functions. So our question here is that whether the presence of coronavirus factors could lead to the hyperinflammation in the host and even cytokine storm by promoting unconventional release of these inflammatory factors. And here we mainly focus on the membrane proteins of the coronaviruses. So to determine this, we first also did a screening on the structure and the non-structural proteins of SARS-CoV-2 by our secretion analysis system. And interestingly, we found that the envelope protein E could greatly boost unconventional secretion, and E-regulated unconventional secretion is not blocked in gastamine D KO cells, indicating that E-regulated unconventional secretion is not through apoptosis. And more interestingly, we find that the E proteins from different human coronavirus showed a different regulation pattern. As shown here, E proteins from severe symptom coronavirus, such as SARS-2, SARS, MERS, could greatly promote unconventional secretion of IL-1 beta, whereas E proteins of mild symptom coronaviruses, including 229E, HKU1, or C43, showed a mild regulation on unconventional secretion. And moreover, by using the mouse inflammation model and AAV to express this E protein in the lung, we found that the presence of E protein of SARS-CoV-2, but not the E protein from the mild strain 229E, the inflammation was greatly enhanced. As you can see that the serum level of IL-1 beta was greatly upregulated, and the inflammation area was enhanced in the presence of E protein of SARS-2, but E protein of 229E showed mild effect on inflammation. So this results indicating that SARS-2 envelope E could promote both unconventional protein secretion and host inflammation. So then we tried to look for the mechanism for E-regulated unconventional secretion, and we found that E protein could co-localize with TMAT10 on ERGIC and could directly associate with TMAT10, and also when we knock down TMAT10 could greatly block E-regulated unconventional secretion, and this secretion could be restored by TMAT10 re-expression, indicating that E-regulated unconventional secretion depends on THU. And in the mouse inflammation model, we found that in TMAT10 KO cells, the presence of E protein of SARS-2 failed to further induce inflammation. So it seemed that indicating that irregulated unconventional secretion and inflammation depends on Th2 pathway. And then we similar to, I think it's pretty similar to the RAP1 proteins. We found that E could facilitate TMAT10 oligomerization and cargo translocation. So indicating that E protein could regulate the inflammatory factor release through Th2, and this could contribute to the coronavirus-induced severe inflammation in the host. And more interestingly, we found that here you could see the interaction pattern of E proteins with TMAT10 is pretty similar to the regulation of unconventional secretion and inflammation by different E's. So here we saw that E protein of SARS-2, SARS, and MERS could strongly associate with TMAT10, whereas E proteins from the mild symptom coronavirus showed a very weak association with TMAT10. So indicating that E and TMAT10 interaction could be a very important regulation site. So then we think if we block E and T10 interaction, whether it will block unconventional secretion and inflammation. So here we use the C-terminal region of E protein, because we found that E and TMAT10 interaction depends on C-terminal region. So here we found that the presence of GLP C-terminal could block E and TMAT10 interaction, and as a result, we saw a strong inhibition of secretion in the presence of GFP ECT. And we further used the mouse coronavirus MHV infection model. We found in the presence of ECT, it could block unconventional secretion and also inhibit MHV induced inflammation. So this indicating that E and TMAT10 interaction is the target for irregulated unconventional protein secretion. So here I would like to summarize this part of work. In this part of work, we identify a molecular mechanism accounting for coronavirus-induced host inflammation. E protein of the severe symptom coronaviruses such as SARS-2, SARS, and MERS strongly interact with TMAT10 to activate the Th2-mediated release of inflammatory cytokines, and therefore explains why severe symptom coronavirus lead to deadly inflammation. And we also identified E and TMAT10 interaction could be a target. And we also find that to reduce in this severe symptom coronavirus, the E protein of this severe symptom coronavirus may be responsible for E and TMAT10 interaction. And this E and T10 as a target could be help us to discover drugs and help us to fight with future maybe future deadly coronaviruses. So here I would like to thank people who did this work. The RAP part of work I collaborate with Dr. Liang Ge at Tsinghua University, and this work is done by my PhD student Tao Xuan, and the work of coronavirus is done by my postdoc Liu Lei and other students Sun Xin, Zhang Lijing, and Hao Xinyan. And thanks the professors Hu Xiaoyu, Ding Qiang, and Tian Boxue at Tsinghua University for their kind help on these projects. And thank our funding source from Wanke Foundation, Tsinghua Dushi Program, and also fundings from NSFC and Key Laboratory of Membrane Biology. So I'll stop here and thank you for your attention. I'm ready for your questions.
Thank you. Very, very interesting talk. So now we're open for questions. Since we have a relatively small audience on Zooms, you can either type your questions in the chat box and I'll read it to Min, or you can actually unmute yourself and just speak out your question. So let's begin with the chat box. And now we have Shenji is asking, actually, these are very interesting questions, which I'm also wondering. When you did your RAB screens, did you find any negative regulating RABs for this unconventional protein secretion pathway?
Negative, we do not see very significant negative regulating RABs. Yeah.
So, and maybe you have it in your slides, but I did
Yeah,
not.
I have my slides
Yeah,
here. Yeah, I think
the screening.
Yeah, some we
The first
saw.
one.
Yes, we saw. That should be in the
very beginning.
Very beginning. Yes. Let me check the. Okay, here.
Yeah. Right.
Yeah. Here we only show the positive one and that ones which do not affect. I'll check. Maybe I'll check later. But we do not see the RAB significantly block this secretion. Yeah.
Yeah, I'm wondering in that regards, did you ever do a siRNA-mediated knockdown screens to look for any RABs when it's knockdown could, could enhance secretion, for example?
Yeah, we did the knockdown to see the enhance of secretion. I think we did some of the RABs, but not all of the RABs. Yeah.
So on this chart, and you also have RAB11 in there, which is a known regulators of unconventional protein secretion, right? Because it's regulating the recycling, but it doesn't seem to have much effects in here, right? Have you tested RAB11 knockdowns in your secretion pathway?
I think we have. It seems that the effect is not obvious. Yeah, it's not like obvious upregulation or downregulation. So yeah.
Yes. All right. So Yuxin in audience asked a very interesting story. So he's wondering how cargoes are specifically selected to go through this unconventional protein secretion pathway.
Yeah, that's a very good question. We for the TMA10-mediated unconventional secretion, we found a residue that is WY motif. It's responsible for it's responsible for the cargo to go through the THU pathway. We mentioned that residue in our previous paper, and I didn't show it in this talk yet. But we have found a potential residue that is responsible for cargo to go through this THU pathway.
So a follow-up on that question, since L1 secretion only occurs after it's mature, right? After the proteolytic cleavage of L1 beta.
Yeah.
And I wonder, you have that motif there, but why it's not working for the immature proteins, but only works when the cleavage happens?
Yes, that's a good question. We do the in vitro translocation assay. Only the L1 beta, mature L1 beta could translocate into the TMA10 proteoliposome. We saw that there's maybe some difference in the structure of pro L1 beta and the mature L1 beta that may be the why pro L1 beta could not go through this pathway.
And have you looked at the structure, published structure of L1 beta to see where that motif is, whether it's actually hidden in the immature protein?
The leading the peptide that the residue that we thought that is responsible for cargo to enter the tier 2 pathways in the N-terminal of the mature L1 beta region. So yeah, but we have yes, for the binding experiment, so except for the in vitro translocation, the other for binding experiment or the other experiment seem that pro I1 beta and the mature I1 beta seem it's the same, but for we could not see pro I1 beta translocate into the T-10 putative liposome. So yeah, that's our guess. Yeah, we haven't checked how about the structures of I1 beta that will make this difference. Yeah.
So let's continue with the questions from the audience. Feng Hua asked, nice talk. Are the activity of RAP1 and RAP2 regulated during inflammations or infection by SARS-CoV-2, for example?
Can you repeat?
Are the activities of RAP1 and 2 regulated during inflation or infection?
Do you mean the the GTP binding activity?
Presumably, right? So the RAP is only active when it's GTP bound forms. In
Yeah,
other words,
yeah.
whether during inflammations you see more RAPs in the GTP form or in GTP form, or how would that be regulated? Is there any evidence?
Here we saw that we show that the GTP binding is required for RAB-regulated unconventional protein secretion. Yeah, but the question is about the inflammation, right?
Right. Yeah. Is it regulated? For example, RNA level or protein level, or maybe the activity of the protein, is it changed during inflammation?
During inflammation.
Yeah.
So here we have this result in the macrophage. We didn't see yes, we did this in the macrophage. We didn't see any. So it still depends on the regulation of RAP proteins in macrophage. The mature I1 beta secretion in macrophage also depends on RAP GTP binding. Yeah.
All right. So Shinji has another question. He's asking, is double membrane vesicle, DMV, where SARS-CoV-2 replicates related to ergic in terms of membrane source?
Relate to ergic in terms of membrane source. Yes. Yes, we think so. Yeah.
Is there any evidence?
I think according to the researchers about the DMV, they saw that the DMV comes from right should comes from ER, right? So and I could not remember, but you can check the paper by, I think the paper by Zhao Yan. Yeah. Yeah, I can send the paper to Shenjie later. Yeah.
All right, Yushin has another question said that does the translocation through TMV data needs energy?
Yeah. If there's energy, it will greatly enhance the translocation activity. Yeah. But since the cargo has a concentration, the concentration of cargo is higher in the out of the the vesicle. So we will so if there's no energy, still can there's still a fraction of cargo can enter into the cargo. Yeah.
So maybe I can ask the last question. So I think there's some evidence in the literature suggesting RAP1, both A and B are also involved in conventional protein secretion, particularly vesicle trafficking between ER and Golgi. But your evidence suggesting that it's exclusively involved in unconventional protein secretion, is that right?
Yes, so that's a very good question. I have, yeah, we have found the mutants. I show you in the slides. In the yeah, here. So we have find these RAM mutants. These mutants do not associate with TMAT10 and also do not have could not promote unconventional protein secretion, but these RAPs could localize this normally on the allergic. So this this indicating that so these three RAP regulated unconventional secretion different from their roles in ER Golgi trafficking. So yeah.
And when the RAP1 binds to TMED10, because I believe TMED10 usually function as a heter oligomer in conventional protein secretions,
Yes.
whereas in your pathway, it needs to dissociate from its heter complex, and then it can be forming this homo oligomer. So I was wondering whether the RAP association occurs before dissociations of TMED10 with its regular partner in conventional secretion pathway, or after that. In other words, does the role of RAP is to help dissociate the normal complex in conventional pathway and therefore promote homo oligomerization, or it's actually doing something after the conventional secretion complex of TMED10 is already disassembled?
Yeah, that's a good question. Yeah, that's yeah. We haven't take a look at, just haven't examined. Take a look at this. I think yeah, we can try to determine whether it is functions before the homo oligomer or hetero limit oligomer forms or dissociates. Yeah, that's a good question.
Thank you. So it's like there's no more questions from the audience, and well, I would like to thank Ming to share her latest work in such a late night and Friday evening. I think we all learned quite a bit. Appreciated that, and hope to see you all in a month. Okay, bye bye.
Okay, thank you, Hong. Bye bye.